So, ladies and gentlemen, hopefully y'all can see my screen. RNA and protein synthesis. If you can't see my screen, now is the time to say something.
We can see your screen.
Awesome sauce. Okay, here we go. Oh, okay. So, ladies and gentlemen, we are going to continue our work with genetics. Now, we've talked for the last 4 weeks, more or less, about DNA. And now we're going to be talking about our other nucleic acid, RNA. So you know that DNA carries out the instructions for the structure and functioning of cells and living things. We know that DNA is on that list of the 7 required things for all living things, and we know the 3 main functions of DNA. The 3 main functions of DNA are to store genetic information, to copy genetic information, and to transmit genetic information, or in other words, to make that genetic information work. So that means, to synthesize the proteins and stuff. But the question is, how are these instructions carried out? How do the genes actually work? And for that, ladies and gentlemen, we are going to take a look for the next couple of weeks at something called RNA. All right, so I'm going to go ahead and do that so you all can go ahead and copy. So we are looking at numbers 1 and 2 in our notes. All right. So genetic code. Again, we know that DNA's job is to store, copy, and then transmit or make that DNA, that information, make it do some kind of work. So now we're trying to figure out, how does it do the work? Because we know that it stores it because of the sequence of the nucleotides. We've spent all of the last almost 10 days talking about how it copies its information. So now we are going to focus on the transmission. How does DNA actually get that genetic information, that genetic code, all of that information? How does it actually work? How do we make a protein? We keep talking about, oh, it makes proteins, it codes for proteins. How do we actually make the proteins? How do we actually make these proteins that make our traits? Okay. So, DNA molecule has 4 nitrogenous bases. We know this, and those code for all the proteins that are in the cell. We know that those genes are made up of DNA. And we know that a gene is just coded DNA instructions, and this controls the specific production of specific proteins. Now, we know that some of our proteins are enzymes, some of our proteins are structural, some of our proteins help carry oxygen throughout the body. So, again, these genes, which is just a bunch of sequences of DNA, are the instructions for building proteins. And then those proteins do different things in our body. Any issues or questions? All right, so this is okay, so we are moving into number 3. So DNA inherited by an organism dictates the synthesis of certain proteins. The word synthesis is just a fancy schmancy word for what you're actually making. So when we say synthesis, that means we're putting something together. We're making something. So the DNA inherited by an organism, remember we talked about heredity, we talked Punnett squares, we talked about Mendel, all those people. So we know that DNA is inherited from parents. The DNA that you inherit determines what proteins you are going to actually make. Going back a little whiles, back to Mendel, we know that proteins are the link between genotype, what is actually in your DNA, and the phenotype, which we know is the expression of those genes. So, for example, myself, I have brown eyes. So, somewhere in my DNA, there is a code of genes, probably on, I'm guessing, well, eye color is actually controlled by several genes, but whatever. One specifically is found on your chromosome number 15. So, somewhere on chromosome 15, there is a code of DNA that set. I'm making this up. I don't know what it really is. AAA, TTT, CGC, for example. And that sequence, AAA, TTT, CGG, means that I end up with a protein that codes for my brown eyes. So the genotype, the actual code, turns into or is visible or is expressed as our phenotype. The proteins that are made are actually going to determine what traits show up in the offspring. Some of you probably have really, really dark brown eyes. Some of you guys might even have hazel eyes. Those are all phenotypes. Each of you, depending on the color of your eye, if your eye color is different, is going to be making a different protein that creates that pigment in your iris, because the iris is the part of the eye that, creates the color. Questions or issues? All right. This is number 4 and number 5 in your notes. So, gene expression, the process by which a DNA directs the synthesis of protein. My gene expression is the process by which my DNA gives instructions to make certain proteins. Like, I don't have blue eyes. So obviously my DNA is not giving any instructions out for that blue pigment, right? Maybe you have hazel eyes. Maybe you are very, very dark skin tone. Maybe you have a very light skin tone. All of those things are proteins in your melanin, which is going to create different shades of color. And those are all just proteins. Now, the expression of gene, the process. No, you're fine. Did you just type it in the wrong spot? It's no worries. Um, the expression of genes happens in 2 stages. So, how do my genes actually get expressed? So, I have all that information in my DNA, all that code, but how do I actually build that protein? The expression of genes happens in 2 stages. Step 1 is transcription, and step 2 is translation. So there are 2 stages, 2 steps to getting from DNA to an actual protein. Transcription followed by translation. Now, maybe over the last couple of weeks, you have noticed that every time I break down what's it called, a strand of DNA, and I'm like, oh, what's the other part of the DNA? I always break it down into threes. I always say AAT space, GGC space, ATA space. I always have these groupings of 3. That was on purpose, because our code is broken down into something that we call triplets. So just like, like you have twins, those are like 2 babies born at the same time, and you have triplets, those are 3 babies born at the same time. Well, a triplet code is a 3 sequence DNA nucleic acid, not nucleic acid, nitrogen base that will code for a specific amino acid. Remember, going back to macromolecules, which was a million years ago, amino acids are the building blocks of proteins. So what DNA really does is it says, hey, we need. Yep, we're still explaining it. So if you have a specific question, feel free to ask. So again, a DNA is going to call up or is going to have instructions that says, "Hey, we need this specific amino acid." Now, there are 20 different amino acids, and there are 4 different nucleotides. This can create literally millions of combinations. Like if you did the math on it, if you assume that you never even had a repeat, I mean, I don't even know. I'd have to actually do the math, but literally there are millions and millions of combinations that can happen here. Because again, my DNA has instructions to go and get part like nucleic acid or amino acids to build the proteins. Okay, we'll get more into this in a minute, but for right now, this is just sort of like the very, very beginning of it. All right. It is discovered that 3 nucleotides in sequence specifies a specific amino acid. Okay, let me explain this. This is like, for example, like like when you go on a website or something and then you type in like, I don't know, I'm just making this up, like, www dot, google.com, it doesn't bring back like the CBS website, right? That code, www.google.com, brings back the Google website. This is a codon chart. And again, if you don't understand it 100% right now, it is not that big of a deal. We'll be working with this all week, okay? But 3 nucleotides are going to be in sequence and they will specify a specific amino acid. Remember, amino acids are the building blocks of proteins. So, this means that there are 64 possible combinations of amino acids. So, again, you have 64 possible amino acids, but of course, we know that there's only 20. So, some of these are going to make the same ones, which is fine. Each triplet of nucleotides is called a codon. So, what are we talking about here? Each codon will call up a specific amino acid. Now, again, amino acids are the building blocks of proteins, which means that once I get enough amino acids together, I have a protein. So, how does this work? When you are reading a codon chart, ladies and gentlemen, you are always going to start in the middle. And again, if you don't 100% understand this right now, it is not a big deal. We'll be working with this all week. In fact, not only just this week, but next week as well. So this is like all the RNA stuff. So let's say, for example, I had adenine, guanine, and guanine again. So I have AGG as a combination, as a codon. Start in the middle, go to the second ring, go to the last ring. AGG brings up the amino acid ARG. I think that's like argonite or argonidium or something like that. Argonase. I forget the actual full name of it. So AGG always start in the center. Remember, because we said 3 nucleotides. Start in the center, go to the outer ring, go to the last ring, and then read the amino acid there. Okay? Again, if you're not 100% understanding this, we will be working on this all week. So don't flip out yet. Issues or questions? Okay. So, a few codons do not call for any amino acids, which is kind of weird, right? But you guys remember like our primase? Okay, like it tells you kind of where to start. We have some amino acids, which are what we call start codons. And a start codon is a specific section of nucleotides where it's exactly like the primase and DNA replication. It says, hey, this is the start of a new protein. Okay. So it's basically a sequence of nucleotides that indicates where the protein building instructions start. There are 3 other codons, which are the stop codons. Those stop codons say, okay, we're done with our protein. So again, your DNA, it's all instructions. We read the DNA in chunks of 3. Those are codons, and the DNA gets turned into proteins in a 2-step process: translation followed by transcription. Some of the stuff that we're talking about here really has the transcription piece, but again, this is just an introduction. We will get into this fully in the next couple of weeks. So again, if you're confused, that is okay, because total introduction day today. Issues or questions so far?
Can you stay on this for this?
What's that?
Can you stay on this for a little bit more?
Can I stay on this slide for a minute? Is that what you said?
Yeah.
Yep, no problem. Let me know when you're ready. All set? Are we ready to move forward?
Yeah.
Okay, here we go.
Yeah.
Okay, nope, fantastic. Oh, I got a no in the chat. We're going to give it one more minute and then we're going to move forward. So let's finish up, please. All right, it's been about a minute. We are going to go ahead and move forward, ladies and gentlemen. All right, so we are now at number 7 in the notes at the bottom of the first page. So a gene of chromosomes is just it's many codons long. Remember, a codon is a grouping of 3: ATT, CGG, whatever, right? It's going to be a grouping of 3. So a gene that is going to make a specific protein is going to be many codons long, because each codon, each grouping of 3, pulls up 1 amino acid. And I literally need like hundreds and hundreds of amino acids to make 1 protein. So please keep that in mind. And again, these genes provide the instructions for making specific proteins. But the gene does not build the protein directly. It's just the instructions. I want you to think of your DNA as like a cookbook. If you are like myself, I'm somebody, I love to cook. I cook all the time. I love to bake. I bake all the time. So whether it's an actual physical cookbook, sometimes I pull instructions up on the internet, it doesn't matter. In front of me, I have this book with instructions, right? That book, though, isn't going to make the cake. No, I have to make the cake. Does that book make the lasagna? No. All I'm doing is I'm reading it to make the lasagna. That is how your DNA works. Your DNA are just provide your DNA, those genes simply provide the instructions for making the protein, but they don't build the protein directly. Just like my cookbook doesn't make the bread directly. Just like my cookbook doesn't make the, enchiladas directly. I have to do that. I just have the instructions that I'm going to follow, if that makes any sense whatsoever. So your DNA is a set of instructions, but it doesn't actually build the protein. Questions or issues?
No questions, Miss Michael.
Okay, moving on. Oh, fudge. So the last part of A right here, ladies and gentlemen, which I, kind of screwed up because I didn't see it on my set of notes. So who actually makes the protein? RNA makes the protein. All right. So just like I said, I have a cookbook. I have to actually gather the ingredients. I have to put those ingredients together. I have to make the lasagna, right? That is what RNA is for DNA. DNA is the instructions, but RNA is going to read the instructions. RNA is going to gather the ingredients. RNA is going to put together the proteins. So just like you might read a recipe and you put together some delicious meal, RNA is the worker who is actually going to do this. DNA is just the instructions. Okay? All right. Oops, went too far. All right, we are on the second page, part A, differences. Yep, it is 8:51. The bell doesn't ring until 8:58 today, just FYI. All right, so there are three main differences between DNA and RNA. Remember, DNA, instructions, RNA is the worker. DNA is the recipe book, RNA is the chef, okay? The recipe book is there, it's got all the instructions, but the chef has to read the instructions, it has to gather all the ingredients, and it actually has to do the work of putting together the meal. DNA is the recipe book, RNA is the chef. Now, what are some differences? Number one, RNA is a single strand. Now, I want you to notice it still has that nice helix shape. It still has that nice spinny, sort of curly shape, but it is not a double helix. It is a single helix. DNA, we know, is a double strand, double helix. All right. Not a big surprise. We've been talking about DNA for like a hot minute now. Issues or questions? Issues or questions going twice? All right. The second main difference between DNA and RNA is that DNA and RNA have different sugars. Remember that nucleotide inside the nucleic acids, right? That nucleotide, it's always like a phosphate group, a sugar group, and then a nitrogen base. That sugar in the sugar group is different. RNA has a sugar called ribose. DNA has a sugar called deoxyribose. I do want to point something out. OSE on the end of a word. If you were in a biology class and you see OSE on the end of a word, you are dealing most of the time with a sugar. Always with a carbohydrate, most of the time with a sugar. So, differences between DNA and RNA. Number one, RNA is single stranded, DNA is double stranded. Number two is the sugar. RNA has ribose as a sugar, DNA has deoxyribose as a sugar. And you can see in the diagram, and I don't think I included this diagram, but I mean, it is slightly chemically different. Deoxyribose here, it's missing an oxygen. I mean, that there's a difference in oxygens in the molecule. All right, third main difference. In DNA, RNA have different nitrogen bases. In RNA, uracil, that is a new one to us. We haven't talked about uracil before. Uracil is going to pair with adenine. Okay, so that's like the new thing going on here. RNA has it does not have thymine. It doesn't have any T's. It has U's instead. DNA, we already know. It has thymine with adenine. That's what we know. We know that. Okay. RNA has a new nitrogen base called uracil, and that uracil pairs up with adenine. I want you to take a look at this Venn diagram. We have DNA over here on one side. We see that it is double-stranded. We see that it has deoxyribose. We see that it has thymine. Those are all unique to DNA. Over here on the RNA side, we have a single strand. We have ribose as a sugar. We have uracil as a unique nitrogen base. What do they have in common? Well, the other bases. By the way, they also have the phosphate group in common as well. But they have adenine, guanine, cytosine 100% in common with each other. They have phosphate groups in common with each other. They have that helix structure in common with each other. Again, one's a double helix, one's a single helix, but that's besides the point. So, three main differences. Number one, DNA is double-stranded, RNA is single-stranded. Number two is the fact that we have, oh my lord, I forgot that. Oh, the sugar, ribose versus deoxyribose. And finally, in RNA, we have this new nitrogen base called uracil, and it is going to do the base pairing with adenine. There is no thymine in RNA. In DNA, of course, we know we have the AT connection. We know that. Here we go, simple thing. If you ever have a test or quiz question that it says anything about RNA and you see thymine in there, you automatically know that one's wrong because RNA doesn't have any thymine. All right, moving on. Where are we on time? We got about 1 more minute. You know what? This is probably a good place to stop because that's the end of that section of notes. So we are going to pause here. So this will obviously go up as part 1 of your notes when I finish up with this video. All right, let me go ahead and stop the recording. Give me 1 second.